We now turn our attention to the research section of our conference. I'm very pleased to welcome Mr. Robert Henderson, consultant ophthalmic surgeon at both Great Ormond Street Hospital and Moorfields Eye Hospital. He'll be sharing some of the latest developments in inherited retinal disease research and what they could mean for people affected. We will also welcome Kate Arkle, our Research Development Manager, who will briefly tell you how Retina UK can support you and your patients with research information. Please join me in welcoming Robert.
Afternoon, everyone, and thank you so much for giving me the time to talk this morning. And it's a real pleasure to see friendly faces. I can see some mums and parents of children that I've looked after over the years in the past, and that is lovely. So. We're quite a mixed audience today, and I recognise a few colleagues of mine, so for them I apologise because a lot of what I'm going to say you either already know, or at least I hope you know. But I'm going to start at a very general base, just to give you a little bit of an idea about what we do in clinic. I think a lot of you already know this, so I will not labour the point too much. And then my aim is to give you a sort of a broader picture of what's happening in the IRD space, really to set us up for question time, because I imagine whenever I do these sort of sessions, the questions I get are much the most interesting part of the talk. So yes, my name's Rob Henderson. I am a paediatric ophthalmologist with an interest in genetic eye disease, particularly inherited retinal disease. But I'm also a paediatric and adult retinal surgeon, and I run the gene therapy service at Great Ormond Street and participate in a number of gene therapy trials. And I will tell you a little bit about what we're doing and a little bit about what's going on in the wider field, as well as we are just starting thanks to Retina UK, we're just starting some early stem cell work as well, which I will give you a little bit of a flavour of. Okay, so I don't think many of you are going to need this, but I always start this talk with just a quick outline of the eye, because often there are allied medical professionals who actually didn't study ophthalmology for any great length of time, and so the nomenclature that we use just needs a little bit of clarification. So the eye, I'm showing a picture of the eye for those of you who can't see my slides, and at the front of the eye we have the cornea, as you know, behind that the anterior chamber, a little space that sits in front of the coloured bit of your eye, your iris. The black dot in the middle is your pupil, and sitting behind that is the lens that does the fine focusing of light. Light then travels through the vitreous cavity, a gel-filled structure, before it impinges on the retina, the photosensitive layer of the eye, the film in the camera, if you like. And the retina turns that light into kind of an electrical signal that travels sideways through the retina, down your optic nerve, and then back to your brain. The joke I always make when I see my kids is assuming they have one. Now, we have lots of ways of measuring vision, and so any of you who've ever been to an eye clinic or work with eye clinics will know that there are an awful lot of different tests that we do, and then the letters that we send out to you contain usually a whole plethora of different information, most of which is jargon and gobbledygook. And so my goal today is to give you a little bit of clarity on some of the nomenclature that we use. But obviously, so when we're looking at patients, we're trying to assess their visual function, we're trying to look at the ocular structures. We have a huge number of, an array of different diagnostic imaging modalities now. And then we can also probe the function of the retina using electrophysiology. So when you get those letters, either you receive them because you're a patient, or you get a letter from me, and you'll know that the first thing I talk about is visual acuity. And yet, visual acuity seems to have any number of different ways of expressing it. We all know from the films, he's got 20/20 vision. But what does that actually mean? What it means is, first of all, that you work in feet, and feet should be banned because they are a ridiculous unit. The same 20/20 is the same as what we say, it's 6/6. So that means at 6 meters, you can see what someone with so-called normal vision can see standing 6 meters away. But if they have 660 or 2200, that means that they can see standing at 20 feet, 6 meters, what someone with normal vision could see standing 200 foot away, or 60 meters. Now, unfortunately, I don't use either of those metrics because I don't use those tests. Those are the old tests that you might remember seeing, where there's one letter at the top and 8 letters at the bottom. And so we don't tend to use those charts, or we try not to use those charts unless you're at Moorfields, which is still based back in the Dark Ages. Ooh, did I say that? I'm so sorry. I do work at both. We're getting a bright, shiny new hospital very soon, in which case everything will be expressed in logmar. Logmar is a proper, sensible unit. So logmar 0 is the same as 2020, is the same as 66, and logmar 1 is the same as 660 or 2200, and you can imagine the scale is somewhere in between. So enough of visual acuities. We also look at visual fields, and so you'll be familiar, I'm sure, many of you, about the types of visual field tests that we do, particularly for inherited retinal disease. And there are 2 main types of visual field. There is so-called static perimetry, which is this one on the left, where a light will shine on or off, and you have to press a buzzer, and hopefully you see it, but sometimes you don't, and gosh, did you see it? And maybe you get trigger happy and start pressing the button a lot. And then there is kinetic visual fields, and this is an old Goldman, which is actually a lovely way of doing it, but it's not terribly objective. And that's where you have a moving target that comes in from the periphery. And we tend to use kinetic perimetry in children because it's slightly easier for kids to get the hang of. And static perimetry tends to be what we tend to do in adults, particularly in the glaucoma population. And then we come to clinical examination. So we have slit lamp exams. You remember all of those. You've had your chin on those, enough of them, or you've seen patients of yours with their chin on them. And we have indirect ophthalmoscopy. I'm going to segue slightly to show you a picture of me examining something on a table, which is, when you pan out, a photograph of me examining a rather larger creature. Now, just to give you a little bit of backstory here, so I was a fellow in Melbourne many years ago now, and I got a phone call while I was in theatre. G'day, is that the ophthalmologist? Yeah, hi. Hi, my name's Rachel from Werribee Zoo. Okay. Listen, you're the local primate expert, and we need you to examine a gorilla. Okay. So would you mind just dropping down sometime? So said gorilla had a heart defect, and they wanted me to look at the eyes. So I said, I'll do that absolutely, providing I can bring my children with me. The thing is, I saw at that exam what I really need in my clinics at Great Ormond Street, which is this gentleman here carrying a large gun. So we have lots of imaging modalities now. One of these is a camera called the Optos camera, invented in Scotland by a physicist whose son was being examined in A&E, and he couldn't believe how primitive our instrumentation was, so he set about creating this unbelievably good camera that allows you to see the entire back of the eye in one picture. And you can image something called autofluorescence, which is what I'm showing here, which tells us which areas of the retina are working, which are making waste products, and which areas are dying or dead. And you can see on the left of the picture, for those of you that can see my slides, very large areas of black, which suggests that there is no metabolic waste products there at all. We have angiography, where we sometimes inject yellow dyes, and we can see what's going on in the blood vessels. We have this technology called optical coherence tomography, which allows us to see at almost cellular level what's going on in the retina. And we can scan through the retina and see structural pictures of the retina, which is incredibly useful when I want to see whether or not your photoreceptors are still there. And now we can actually image in real time what's going on in the blood vessels and whether or not those blood vessels are open or closed, and we can do that using the same technology. And in fact, the technology is changing dramatically all the time. It's extraordinary. And then finally, many of you will know or remember, if you've had it, electrophysiology, where they stick these little sticky electrodes on your cheek if you're a child, or they put wires in your eyes and wires on the back of your head and flash all sorts of unpleasant lights at you. And we get an idea of how the retina is actually working. So we have an electroretinogram and a visual evoked potential. And that's an incredibly useful way to make a diagnosis of a retinal dystrophy. So we move on now away from that to the genetics of inherited retinal disease. And as I'm sure you're all familiar, there are a massive number, 270, maybe even 280 genes that can cause a retinal dystrophy, and they have overlapping functions. So you have genes that can cause both Leber's congenital amaurosis, and it can also cause retinitis pigmentosa, and you can have other conditions like Bardet-Biedl syndrome, which will have a wide range of syndromic effects. So there are a lot of overlapping genes that cause overlapping conditions, and the genetics has not been entirely straightforward, and we're still finding new genes all the time. So the world of diagnostics has changed dramatically, both with our ability to image, but also the type of genetic testing that we're doing. So you may remember a little while ago we had the Human Genome Project, and that allowed us to decode the whole genome. And then one of the great things that the last Cameron government did was this pilot called the 100,000 Genome Project, and that led to the state of play as we have it at the moment, whereby if you walk into my clinic, or theoretically any eye clinic in the land, and you present with signs of an inherited retinal or an inherited eye disease, I as a clinician should be able to ask NHS England to do a whole genome test for pretty much any inherited eye disease, which is extraordinary. So the cost of doing genetic testing has fallen through the floor. So when I was doing research back in the dark ages, it feels like now, we would take an individual patient's DNA, and I would want to look at one specific gene, and I would have to, doing a bit of witchcraft and wizardry, I would have to take that DNA and I would do something called sequencing. And what I'm showing on the slide at the moment is something called an electropherogram, which is all of the A's and T's and C's and G's that make up your genetic code. And I'd have to go through each individual piece of each individual gene. Well, you can imagine that just took forever. So you'd concentrate in your research on maybe one or two genes. But now, what we're able to do is not just look at one gene, not look at, hundreds of genes in one go. I can look in one experiment at all 26,000 genes. And that whole process takes 24 hours to do an analysis, and we're left with, well, several CDs worth of information. Actually, no one uses CDs anymore, do they? But the point is, a vast amount of information. Now, in truth, we're not really interested in an eye clinic at looking at every single one of your 26,000 genes. That would take rather too long. So we tend to refine it down to the genes we know, and then it goes through what we call an analysis pipeline, and then a year later, which is what the turnaround time is at the moment, it's a bit embarrassing, but we get hopefully an answer. And we tend to find an answer for inherited retinal disease about 70% of the time. So now that we know your gene, what can I do with that information? Well, part of it's really useful, because if I have seen someone with that gene before, hopefully I can give you either the professionals working with patients, or I can give you, the patient, a better idea of what that looks like for the future, what we've seen in larger cohorts. And so certainly for the last 10 or 20 years, we've been working up cohorts of patients, trying to establish what we call the natural history of disease. I wish it was as straightforward as you have one gene and you have one, what we call phenotype, but unfortunately what we've discovered is that genes can be unbelievably variable, and depending on what type of mutation you have, may determine whether or not you have a mild version of RP or might have a really bad version of RP. So it hasn't proven quite as straightforward as being able to say, you've got CRB1 and that means you're going to have this outcome. We've actually worked out that there are probably 10 different discrete outcomes that you can see with CRB1. But nevertheless, it does hold out the hope that if I know what your gene is, I might be able to treat it. And so now we get onto the bit that you've actually all been waiting for, which is actually what are you going to do with all of this? Where's all the research going? And so the first thing I'm going to tell you about is gene therapy. So gene therapy, the idea behind it is that I, simply put, can put a healthy copy of the gene that you are missing or that is mutated into the cells of the retina, and then we hope the retina will start working again. How do we get that information in there? Well, we at the moment, use viruses. Viruses are specialist instruments for posting DNA into cells. That's what they were put on the earth to do. And they don't do much else, truthfully. What they do is inject, you can imagine, a cold virus. You get sneezed upon. That hot cold virus hits your nasopharynx. It posts the cold virus DNA into your cells. Your cells goes, ooh, look, a new set of instructions. Let's make lots more cold virus. At which point, the cell splits open and you sneeze and sneeze out 1 billion more cold viruses at someone else. So obviously, I don't want to infect you with anything. So what I do is that we take a virus that doesn't cause disease in humans. So the one that we typically use in eyes is a virus called the adeno-associated virus. And we take out that virus's own DNA and we insert into the virus the copy of the gene that we want expressed in the cells of the retina. And then we inject that either into the eye in what's called an intravitreal injection, and that goes into the jelly cavity, or we do an operation, and I'll show you if you're not feeling too squeamish, an operation where I inject under the retina a bazillion copies of this virus, and then those cells copy themselves, those viruses copy themselves into the cells, and things start to work. So here's a schematic essentially outlining what we're trying to do. You can see, for those of you looking at my slides at the bottom, this is some of the work we do when we're preparing gene therapies. We're trying to get to see whether they work. We want to know which cells of the retina are actually being so-called transfected by this virus. And so the way we find that out is that we put a green fluorescent jellyfish protein into the virus. And you can see, this is a picture of the whole retina, and you can see that there is green lighting up bits of the retina that have been transfected. So we know that this particular virus is really good at transfecting photoreceptors. It's a little bit less good at transfecting bipolar cells which are coming up, and it's hopeless at transfecting the ganglion cells of the inner side here. So that's really useful. So that's how we start to work out which type of viruses we want. So here is a video of me treating using the only licensed gene therapy that we currently have, which is called Luxturna, or voretigene neparvovec. I'm sure most of you know that there is one licensed gene therapy, and it's for the treatment of an early onset retinal dystrophy caused by a gene known as RPE65. And so what I'm showing with this video is a picture of, or in fact a video of me injecting, forming a blister of fluid under the retina. On the right-hand side, you can see intraoperative OCT. So I can visualize using an OCT built into my microscope, making sure that I'm injecting it in exactly the right place. Because sometimes the fluid doesn't always behave itself, and so it goes in places that I don't necessarily always want it to. So having an OCT is really important. Now, what I'm going to show now is videos of a little boy who I treated at the age of 2. His mum's a wonderful person. She allows me to show this video everywhere I go because he is probably the most dramatic example of how powerful this treatment is. So, I don't know if the sound is going to come through. Here we go. So,
where's the door to the room, Harry?
His father is asking him where the door to the room is.
Where's the door to the room?
He is standing by his cot. He's
cot.
feeling his way down the side of the cot. And this is a room with normal bedroom overhead lighting.
Yeah.
Where's the room door?
And his father keeps whispering, where's the room door? And he's feeling, and he's got to the end of the room, and he's feeling the wall. He is very clearly a child with very significant visual impairment. He looks up at the light because these RP65 children love light. They love light. It's about the only time when you're out in bright sunlight they can sort of function.
There, well done.
And he's finally found his way to the door. I'm now going to show you him about 9 months later, after gene therapy in both eyes. He's kneeling on the floor, and he's picking up polystyrene beads from his beanbag off the floor. It's absolutely extraordinary. Now, I can't tell you that every patient who is treated with with Luxturna has this dramatic improvement. Part of this is down to the fact that most people with early onset retinal dystrophies have never really learnt to see. So there are two parts of vision. There is the eye that turns the light into an electrical signal, but then there is the brain that is learning to take that signal and learning what to do with it, learning to anchor you in place, learning how to reach out and grab things, to interpret the world, sending off projections to all the other areas of the brain. And that learning happens in the first few years of life. And if you don't have that learning, even if I were able to take your eyes out and put a new pair in that work normally, your brain wouldn't know what to do with it. We call this amblyopia. And you've often seen children wearing patches over one eye, and that's because we're trying to get them to strengthen the eye that's uncovered. And so you can have bilateral amblyopia, and most children who I treat who are older don't get this massive improvement in vision because they are bilaterally amblyopic. And that's probably one of the reasons why some of these trials that we're seeing and doing fail. So, for example, for those of your patients who may have something called achromatopsia. Achromatopsia is a condition where your cone cells are not programmed to be able to work quite properly. Cone cells are your colour vision cells, your daytime vision cells, the cells that give you detail. And patients with achromatopsia dislike bright light because their cone cells aren't working well. Their daytime vision is not great. Their colour vision is absent. So we had a gene therapy trial. In fact, we've had several gene therapy trials for CNGB3, one of the genes for achromatopsia, which did absolutely nothing. And everyone was rather puzzled because all the animal models looked rather good. And yet there was a really interesting little subpaper that showed that in some of these patients, if you looked at functional MRI, their visual pathways lit up. There was clearly signal suddenly happening, but the brain just didn't know what to do with it. And the trouble is, all of these patients who were being treated were far too old. So it's possible that actually, if we treated achromatopsia when a patient was 1, we might be able to do something. But there aren't many people who are happy operating on 1-year-olds. So what are the current drawbacks? Well, and I'm probably running horribly over time, aren't I? Oh, God. Okay, I'll whistle. 10 minutes. Really? Oh, God. Okay. So one of the problems we have is that the adeno-associated virus can only take smaller genes. So there are viruses that can take bigger genes, but they unfortunately cause disease in humans, or we have we're likely to develop an immune reaction to them. So there are difficulties at the moment. One of the ways we get round that is to split a gene into 2 parts. So there is currently a trial that I'm running over at Moorfields with our colleagues down in Naples in Italy, looking at the myosin 7A gene. It's a gene that causes type 1 Usher syndrome. And what they've done is split the gene into 2 pieces, put both of those pieces into individual viruses, and then we inject both of those viruses under the retina, and a proportion of those 2 pieces will rejoin back in the cells again and hopefully start working. So that's an exciting new strategy, and actually that trial, that company are also launching a trial for a similarly big gene for Stargardt disease, the ABCA4 gene. So moving away from gene therapy, we have optogenetics. Optogenetics is a fascinating area. The idea is that if you've got no functioning photoreceptors and gene therapy isn't going to bring cells back to life, how are you going to turn light into an electrical signal? Well, how about we take, those anglerfish that sit at the bottom of the ocean, the ones that have a little dangly thing over an enormous jaw? Well, they have different types of what are called opsins. And maybe if you can take one of those type of opsins, a molecule that turns light into an electrical signal, and you put that into the inner retina, maybe you could get the cells of the retina to start firing by using one of these proteins instead of one's own opsins in one's own photoreceptors. And so that is the basis of optogenetics. And there are now phase 3 trials for optogenetics that make some extraordinary claims. Most of us are still scratching our heads, not quite able to believe that they're actually getting these results. So I take a slightly sceptical tone with it because I'm not entirely convinced. Nevertheless, there is quite a lot of room for excitement that optogenetics may be a cure for people who have lost all of the photoreceptors. What about stem cells? So everyone's heard of stem cells. Stem cells are essentially, for those of you that don't know what a stem cell is a cell that you can differentiate from into any type of tissue you want if you give it the right molecular cues. You can get stem cells from human embryos. You can get stem cells from the umbilical cord. You can get stem cells even from the eye. And even more remarkably now, I can take a little skin snip from you, I can reverse engineer it into a stem cell, and then re-engineer it and turn it into a photoreceptor cell. So it is genuinely amazing what we're able to do. So to give you a little hint of some of the work that we're currently trying to do, so my colleague Jane Sowden, and you'll hear from her tomorrow if you're here tomorrow, she and I are working on a project where she in the lab can grow up something called an eye cup. An eye cup is like a little mini diddly eye in a little dish. In fact, she can grow up hundreds of these little tiny little eye cups. And then she can dissect out the retina from these little eye cups and sort the rods and the cones. And then she will print at a micron scale a little structure into which she will seed these rods and cones at a density almost approaching that of the human retina. And then the idea is that we will hopefully, once we've got through our animal studies, be able to put these into the retina of patients who have lost their rods and cones with the hope that the cells will grow out of that little ultrastructure that we've created and form synapses and start firing. So enormously exciting work. It's a long way away. I'm not going to try and pretend that's around the corner. But we are now starting, at least in the animal models, and it's thanks to Retina UK that we're able to do that. So finally, just to give you an idea of some of the trials that are currently running in the UK at this particular time, X-linked RP has actually just finished recruiting. There were 2 trials, one of which you may or may not know this. There were 2 trials. Mira GTX, that was originally a spin-out company from Moorfields, had a phase 3 trial for X-linked RP. That unfortunately did not meet its primary endpoint, so that has terminated and won't go any further. Beacon Therapeutics, which those of you that know of Rob McLaren in Oxford, he managed to convince the Wellcome Trust spin-out arm called Syncona to go off and buy a company in the US. And so he now has a new plaything. And this has a phase 3 trial that has just finished recruitment for X-linked RP, and we're crossing all our fingers and toes that is going to come good. And that's really exciting. I'm currently coming to the end of a trial for a condition called Batten's disease, CLN2 Batten's disease, which is a devastating pediatric disease. That's got incredible data, so I'm really excited about that. I'm about to start a trial for Bardet-Biedl syndrome type 1. So at Great Ormond Street and at St Thomas's together, we will be starting a phase 1-2 trial for BBS1. We're in the process at the moment of USH1, as I highlighted earlier, over at Moorfields and in Naples. And there will be a trial coming up for ABCA4. And then there are a couple of other trials, both for USH2A. These are all, I'm just throwing this out there for information, just USH2A and CEP290, which is a slightly different technology. I think that's enough. I've completely overrun. I'm so sorry, Kate. I will hand over to you.
Thank you very much. Thank you so much, Rob, for that really enjoyable overview of what's going on in research. I'm going to be very quick. I don't have any gorillas to talk about, so it's really helpful to be up to date with what's going on, and having that context, I think, gives us all confidence when we're speaking to people affected by these conditions. Lots of our community are really well informed. They go away and they find out about things, they want to talk about them, or they might just raise their hopes and fears for the future when they're chatting to you. And I think it's really helpful to have some idea of where the science is going. However, I know that you're all incredibly busy. It would be really hard to always keep completely up to date with all the clinical trials, all the research developments, and you don't always have an awful lot of time with your patients and clients. So that's where we can help you out. We're really proud that the survey this year, 81% of our respondents said that they were better informed about ongoing research because of their contact with Retina UK. And since we started our surveys, a clear majority of people have consistently said that we're their go-to source of research information. So I'm really proud of that. We report on research developments via the research news section of our website, and we then share those via our social media channels and in our e-newsletters and our main newsletter. So please do encourage people to sign up for those. We also have amongst our resources on our website some podcasts and webinars that go into more in-depth dives onto various research topics, and occasionally we also have Ask the Experts sessions where people can come and ask somebody like Rob all their burning questions about research, and the recordings of those are on the website. You're also really welcome to get in direct contact with us, so those you're supporting as well, with research queries. So the helpline are really happy to pass queries on to me or my colleague Eve, who's our research coordinator, or you can just get in touch with head office, and my email address and Eve's email address are in the delegate list for today, so please don't hesitate in just asking questions. Rob mentioned genetics, and earlier today, Tina, I think, mentioned unlocked genetics. Do bear that in mind. Genetic diagnosis is so important, and there's lots of easy to understand information in unlocked genetics about genes, inheritance, what genetic testing involves, what genetic counselling is, and how to access those things. So please do signpost people to that. And hopefully they will feel more empowered to go seeking those services if they want to do so. Our survey this year also revealed that while a quarter of respondents had taken part in some form of research, more than half of them wanted to but hadn't yet been able to. So we're really pleased that research participation is built into the new eye care support pathway. And we can also help out. Anyone with an inherited retinal condition can join our lived experience panel, and we will then email them anytime we hear about an opportunity for them to take part in a research study. Those are occasionally clinical trials, but there are all kinds of research that relate to life with sight loss. Sometimes there's surveys and interviews that can even be done from home. You can sign up, or people can sign up to our lived experience panel on our website, or just call the helpline or the office, and we can help them out with that. If you yourself are involved in running a study and you would like any help with PPI or participant recruitment, then please do just get in touch with me or Eve, and we can have a chat with you about your study and share details. And as Rob's mentioned, as has been mentioned earlier today as well, we do also fund research. Rob and Jane's project is really, really exciting. Jane is going to be talking about that in some detail tomorrow. So join us online if you're not coming back in person. And we have lots of other really exciting projects going on as well. And you can read about all of those on our website. And bear in mind, we can only do this research with support from our fantastic donors. So again, a huge thank you to Rob for giving us that overview today. Rob, I'm sure some of you will have some questions. So have we got, really? Okay, yeah, we've run out of time. Sorry. Okay, but please do send me your questions, send Eve your questions. We can always ask Rob if we need to. We'll do our best to help you. I think Rob's got to run off again, but I'm around if you have any questions, and we'll do our best. Thank you.